In this tutorial, I will introduce you a new software tool called XMapper. XMapper is a software tool used to map and score the crosslinks or data bonds in proteins and peptides by use of mass matrix database search results. The results are displayed in color maps called XMaps and also in Excel spreadsheets, which you can open for further analysis. Okay, this is an example of mass matrix search results with data bound search function enabled. So you can see that you will have a long list of peptides with crosslinks or data bounds, and also then you have to go through the whole list, try to assign one or two crosslinks or data bounds for each peptide, and then you'll have to compile all those crosslinks or data bounds. And then you may want something like this, a XMap. XMap is a scaled color map, which is used to represent all the crosslinks or data bounds in a protein. For example, this is the XMap for data bounds in ribonuclease A. And a XMap contains many cells, like this one. Each cell represents a crosslink or data bound in that protein. For example, this cell represents the data bound between cysteine 26 and cysteine 40, and this cell represents data bounds between data bound between cysteine 50 and 100. And the color of the cells tells you the score of those crosslinks, and this is a scale which lets you correlate those colors in the XMap to the actual scores of those crosslinks. Next, I'm going to demonstrate how to install XMapper on your computer. In order to install XMapper on your computer, first you have to download the program. And you can go to the official website at UIC or the official website of Mass Matrix at OSU to download XMapper. You go to the search.rc.uic.edu, go to the download site, and scroll down the web page, and at the end, the last program is the XMapper, which contains the links that's going to direct you to the download page of XMapper, and also it contains links to this video tutorial. And I'm not going to download the program because I already did. I'm going to just demonstrate how to install the program. So after you download the program, you double click the installation package, and then follow the instructions in the setup wizard. It's very easy. You can just accept all the default settings and go all the way to the end, and it's going to take a while, so it's going to take a while. In the meantime, so after this, the installation will be finished, and you're done. So in the meantime, let me explain a little bit about how to use. Let me explain a little bit more about XMapper. So these are the two examples about the XMappers for two proteins actually. The first one is the XMap for data bound of insulin. We know that insulin contains two chains. Let's take a look at the insulin contains two chains, okay? And for this type of the XMaps, actually, if you have a protein with different chains, okay, now it's finished. And if it's finished, then after you install the program, there will be icon on your desktop. This is an XMapper icon on your desktop. And let's go back to installation and uninstallation. After you install the program, if you want to uninstall the program, you go to Start, All Programs, XMapper, and uninstall. Click this. And it's going to completely remove the program from your computer. So this is installation and uninstallation. Next, I'm going to demonstrate how to use the program. It's actually very easy. So first step is that you have to download the Mass Matrix search results from Mass Matrix server and unzip the results. If you have the old Mass Matrix PC version, then you probably don't have to do that because those results are local. After you download those results and unzip those results, then you can use the XMapper. So when you install the XMapper on your computer, actually, there are example data coming along with the installation. So this is the example data. It contains 3 sets of mass matrix search results. Okay, I'm going to analyze the 3 at once. So you can actually choose 1 or more mass matrix search results. I'm going to choose all 3 and grab those results to the program, XMapper program. Okay, and then XMapper is gonna process those mass matrix search results one by one. And for each file, I will ask you the maximum scale of the color map. For the first time when you use it, if you don't know anything about it, you just skip this by press enter and accept the default value. Okay, now it's finished. After it's finished, for each protein match in each mass matrix search file, there will be 1 PNG file generated and 1 spreadsheet CSV file generated. And each PNG file contains the XMap for one specific protein in the search result. Okay, so that's very easy to use it. That's it about how to use the program. Next, I'm going to talk about a little bit more about the interpretation of those XMaps. So these are the 2 XMaps for 2 proteins. This is the data bounds for insulin. And like I said, all the cells, it contains many cells, and the cells are separated by thin white bars. And you can see that for this one, it also contains 2 bold white bars. These 2 bold white bars are used to separate 2 chains. Okay, so this represents all the data bounds within chain A. This section represents all the data bounds between A and B, chain A and chain B. This section contains all the data bounds, which is only 1, within chain B. And this is another type of XMap. This is XMap for crosslinks between cysteine and lysine, because this is crosslinks between 2 different amino acid residues. So the XMap will be rectangle instead of triangle, because one cysteine. Because if you're talking about like data bounds, cysteine 7 to cysteine 5 is the same data bond as the cysteine 5 to 7, so it's only rectangle. But when you're talking about the crosslinks between 2 different amino acid residues, then we're looking at a rectangle like this. And also you have the bold white bars. You have 2 of them, which are used to separate different chains. Actually, this one we are looking at the crosslink between 2 different proteins. So it separates 2 different proteins. So this section represents all the crosslinks within protein A. This section represents all the this section, and this section represents all the crosslinks between A and B. This section represents all the crosslinks within B. And of course, each cell represents a single crosslink labeled by between its row label and column label, and the color of the cell represents its score. So like this cell represents the crosslink between cysteine 93 on chain B and lysine 35 on chain A, and its score is around 20. So, and that's it. And this is context. If you have any questions, you can email me or attach any comments to this video. Thank you for watching. Thank you. Bye.